In this video, we are going to talk about biofilms. This video is brought to you by Group 5, composed of my friends Tae Kai-sen,
Hi.
Soyan Ru,
Hello.
Su Shali, Scott Audrey Paul, and I, Dan Hao Dong. So let us begin. What are biofilms? Well, they are a type of bacterial colony in which communities of bacteria are formed. A distinction between biofilm bacteria and planktonic or normal singular bacteria is the green slimy substance that holds biofilm bacteria together. This substance is composed of exopolysaccharides and can account for 90% biofilm mass. Most often, these biofilms are composed of an array of different bacterial species, but they can be of a singular species as well. It's been increasingly acknowledged that bacteria actually spend most of their time existing in biofilms rather than wafting alone. Well, where are they commonly located? They are actually everywhere, from the plexus of teeth to our gastrointestinal tract.
Yes, they are everywhere, including on abiotic surfaces. For example, supermarket trolley handle. A lot of Staphylococcus aureus biofilm can be found. In fact, when we touch abiotic surfaces, planktonic cells can survive up to 3 hours on our hands. Interestingly, after we use bathroom without washing hands, the number of germs on our fingertips will be double out. What would happen if we let this pathogen be? Well, most of them will die eventually, but a small percentage of them will contaminate our food. The table is showing that the duration of persistence on our hands is different in various pathogens. Besides contamination of food and beverage by hand, they can enter our body due to contamination in the food industries. Let's take E. coli and Salmonella for examples. Both E. coli and Salmonella can form biofilm. What would happen if we swallow them?
Well, a lot of things can be happen. It depends on the bacteria. Example, pathology of Salmonella is by ingestion in food survive passage through gastric acid barrier and invading the mucosa of small and large intestine to produce toxin. The epithelial cell stimulates the release of pro-inflammatory cytokine, which causes an inflammatory reaction, as this reaction causes diarrhea and ulceration and destruction of mucosa. The bacteria could cause systemic disease such as red dot and red streak. For E. coli, it produces Zika-like toxin and causes hemolytic uremic syndrome, which absorbs through inflamed gastrointestinal mucosa into circulation and alters the function of endothelial cell, which leads to platelet activation and aggression. In results, children and elderly are at high risk.
But not all biofilms are bad. In general, microbes have a bad reputation as disease-causing germs, but most microbes are essential for the survival and well-being of many organisms, especially those that reside in the biofilm in human and animals. One of the significance of microorganisms in the biofilm is these healthy cohabitants contribute to the well-being of marine mammals by reducing the risk of infection. Some microbes can produce antimicrobial molecules. They can be fatty acids, peroxides, and bacteriocins, which inhibit or kill foreign bacteria. Such good microbes in the biofilm is sufficient to deter unwanted harmful microbes. Therefore, commensal microbiota in the biofilm acts as a line of defense and protect marine mammals from potential microorganisms that cause disease. Another significance is that they participate in metabolism. For example, let's talk about wax ester. Wax ester is one of the most difficult fats to digest for many animals, including humans. They are especially rich in tiny crustaceans such as krill and copepods, which is a favorite prey for bowhead whales. Malabsorption of these lipids can lead to diarrhea and bloating. However, a study has shown that the gut bacteria in the biofilm within whales help the whales break down these fatty prey compounds. It is known that more than 80% of the lipid consumed by the whales are wax ester, but less than 30% remain in the large intestines. Researchers collected 100 samples from 9 locations along the gastrointestinal tract of 38 bowhead whales over 4 years. They found a strong connection between the bacterial community in the biofilm and a decrease in the amount of wax ester in the lower part of the small intestines.
Yes, be it for the betterment of our health or the worsening of it, bacteria impacts our lives indefinitely. But what exactly are biofilms? How do they form? Well, let's take a look at the diagram. As you can see, it all starts when a planktonic bacteria, once wafting around in the air or water, coming into contact with a surface, conveniently called the substratum. Once the bacteria deems the surface to be suitable, it proceeds to attach itself to the surface using various bonds. Once firmly attached, what happens next is that the cells start to communicate to each other using a process called quorum sensing. It works by a mechanism of signal molecules which increases with cell density. At specific concentration of this family of chemicals, it can control various aspects of the bacteria's activity, including growth, virulence, and of course, biofilm forming factors. In the end, a tenacious three-dimensional gelatinous matrix is formed, adhering the bacteria onto the surface with limited motility but extreme resilience.
So, how is the biofilm formed in mammalian gut? The traditional view of gastrointestinal tract of a normal fetus is sterile, and microbial inoculation occurring during parturition has been challenged in the past few years by multiple lines of evidence. This evidence suggested that bacteria are actually present in the intrauterine environment, which basically refers into the uterus. Generally, the first colonizers in the gut are facultative anaerobes, for example, enterococci and enterobacteria, followed by obligate anaerobes like Bacteroides, actinobacteria, and Firmicutes. In human, a gut flora and intestinal biofilm similar to an adult's is formed within 1 to 2 years of birth. Some sources of bacteria in the biofilm includes the birth canal, parent-to-children transmission, other people such as relative and hospital workers, breast milk, food, pets, and the general environment which the infant interacts with. Cesarean section, antibiotics, and formula feeding may alter the biofilm composition in babies. Children treated with antibiotic lack of a healthy and diverse floral community in their biofilm. Cesarean section has shown to disrupt the transmission of bacteria from mother to offspring. This impacts the overall health of children by increasing the risk of diseases such as celiac disease and asthma. Celiac disease is also known as a gluten-sensitive enteropathy. It is an immune reaction when consuming gluten that is found in wheat, barley, and rye. Asthma is a chronic airway condition in which the airway narrows, swell, and produce extra mucus that leads to difficulties in breathing. Other than that, biofilm of the breastfed babies are dominated by bifidobacteria. This may be due to the bifidobacterial growth factor and probiotic components in the breast milk. In contrast, the biofilm of formula-fed infants are more diverse, with higher numbers of Enterobacteriaceae, Enterococci, bifidobacteria, Bacteroides, and Clostridia.
An implementation of biofilms can be said to be the stone that kills two birds, and here's why. Biofilms are a solution not just for wastewater treatment, but for renewable energy as well. This can be achieved through a device known as the microbial fuel cell. A microbial fuel cell is a type of bioelectrochemical system, otherwise known as biofuels. It's a device that uses biocatalysts such as bacteria to convert chemical energy into electrical energy. Here's a little more on how an MFC works. It's much like a battery cell, but using bacteria to break down nutrients which produces electrons from the anode to the cathode. The anode is placed in an anaerobic media to control the binding between oxygen, to encourage the electron transfer from the anode to the cathode. Electron transfer is what generates the energy in the MFC. Many challenges arises in an MFC. The bioelectric capabilities of the bacteria is one of them. To date, only a few have been isolated in pure cultures with the ability to produce a high current density. One such species would be the Geobacter sulphurinducens. It's speculated to form an electrical conductive biofilm capable of electron transfer from a considerable distance greater than 50 micrometers. To compensate the distance, energy generated is increased by the addition of electron acceptors into the MFC. For example, potassium ferrocyanide and manganese oxide. These electron acceptors were used to attach to the inner mitochondrial membranes or cell bodies of the bacteria by saturating the electrodes. This, in turn, increases the energy generated. Biofilm on an industrial scale. Engineering is a challenge when talking about MFCs in wastewater treatment. The desired characteristics or materials used are crucial. For example, electrical conductivity, corrosion resistant, eco-friendly and low cost. Materials that are commonly used are the carbon veil, carbon brush or carbonized cardboard with high electrical conductivity. From a researcher using laboratory MFCs, the MFC managed to reduce inorganic pollutants by 84%. Now that is effective wastewater treatment. The same MFC produced 250 milliwatts. From this, it can be said that a power plant with an MFC generates 3 times the energy needed to run the plant. Let's talk in terms of business. In a month, the power plant can make $220,650, which basically means it can cover the operation cost for close to 2 years. Not to forget, the plant uses 1/3 of the energy created, which means the leftover energy can be used to power homes. 820 homes per year, that is.
We have approached the end of our video. In the end, bacteria are fascinating. All this time, we have been underestimating their roles in our lives, as well as the potential contribution they can provide. Hopefully, in a future not too distant, we will be able to properly utilize bacteria in terms of biofilm control to make our world a better place. Thank you.